The broadcast is now starting. All attendees are in listen-only mode.
Good afternoon, everyone. I'm Kirsten Wakefield. I'm the Stakeholder Outreach Liaison for AmeriCus and NECAN. We're really excited to have Dr. Amy Maas and Dr. Emily Rvest join us today to discuss their work in developing biological indicators to better understand the ecological impacts of ocean acidification in open and nearshore waters. Co-locating biological and chemical monitoring has been identified as an area of need in both the Mid-Atlantic and our neighboring coastal acidification networks. This webinar is one of the first steps we're taking to explore these linkages and how they might be integrated into a regional monitoring network. So today we're pleased to host this webinar in partnership with NECAN and SOCAN, the Northeast and Southeast Coastal Acidification Networks. Janet and Sam, would you like to say a few quick words about your coastal acidification networks? I'll turn it over to Janet.
Janet, you may be on mute.
I am. I'm really sorry. You'd think we'd all learn by now. Thanks again, Kirsten. We are very excited to be able to share with your membership and as well with NECAN some of the things that are going on in the Southeast region. So just to give you a really quick update on a couple of projects and introduce the SOCAN team. So we are hosted through SOCORA, which is the Regional IUCE Association for the Southeast region. And we're very grateful that we get to overlap with GCAN, the Gulf of Mexico, as we acidification network, because we share the West Florida shelf. Deborah Hernandez is the executive director of SOCORA, which is our fiscal home. Laura Corman is our SOCORA liaison and has been also helping us on most of our social media. Emily Hall from Moat Marine Lab and myself were the program co-coordinators. If anybody would like to learn more about SOCAN or how you can be involved, even if you're not part of the Southeast region, we'd still really love to hear from you. You can email us at socan@socora.org and follow us on Facebook and Twitter. Mostly what we do is host virtual meetings and webinars and workshops these days, but we've actually found that it's been to our advantage because we're able to get more participation because our region is so large. And I'm going to highlight today 4 projects that are occurring in our region. Oh, sorry. So to start, we're very research heavy in Florida, much more so than in South Carolina, North Carolina, and Georgia. And that we think that that mostly has to do with the fact that socioeconomically, there's been a larger impact in Florida than we've seen yet in South Carolina, North Carolina, and Georgia. Some things that are going on, we have a lot of red tide research. Emily, my SOCAN co-coordinator, is PI on a project right now with Kate Hubbard at the Florida FWRI. You're gonna have to excuse me, I completely am drawing a blank on what it stands for. But they are looking at Karenia brevis red tide blooms and carbonate variables. And early results show that Karenia brevis likes lower pH. So with increasing acidification, we might expect to see more red tide blooms, especially in Florida. And then we can move over to Tampa Bay, who's leading the research in seagrass restoration and co-locating carbonate variables and seagrass counts, because as Tampa Bay was eutrophied in between the 1970s and 1980s, they lost a lot of their seagrass beds and thus a lot of habitat. Once seagrass beds were restored, interestingly, pH started to increase in Tampa Bay again. It's been a much slower restoration process than it was like a decreasing, where when seagrass beds decreased a lot faster than they've been increasing, but we've seen a steady increase over the last about 20 years since much more stringent practices were put into place for wastewater treatment and mitigation of nutrients throughout the bay. Another project that is going on is actually just started this year is the coastal acidification research in South Carolina. This project is actually co-funded. It's funded through South Carolina Sea Grant, and SOCAN is actually a co-PI instead of just, you know, it being a project that's occurring in our region. We are one of the PIs on the project. We're going to be looking at historical data from YSI SONs and hopefully being able to validate that information to be able to use it in coastal estuaries where we don't need as high precision as we would maybe in the open ocean. So that data, the data that's being collected there will start this year with carbonate system variables, and then we're going to go back and use pH, historical pH, chlorophyll, and nutrients, but continue using those because those are leveraged variables. And then finally, the only project in the region that we know of right now that actually uses citizen science sample collection is the Georgia Coastal Ecosystems Project. And that sample collection, the citizen science sample collection, is occurring up in the Altamaha River. For those of you that are not as familiar maybe with the Southeast, the Altamaha River is the largest watershed in the Southeast region. As you can see, it goes up through about a third of Georgia. Those samples have been collected since 2013, and some initial results that I can share with you today is that we do see hotspots for acidification, which would be the low aragonite saturation states within the coastal marshes. You can particularly see them in May, July, November, and December. It's that blue color. So we're very excited to be co-sponsoring some of these projects and collaborating with others. So if you're interested in learning more about what's happening in the Southeast region, signing up for our monthly emails will keep you informed of different projects and opportunities for participation in our upcoming research that will be, we have a project funded through OAP, the Ocean Acidification Program, trying to find our priority monitoring sites throughout the Southeast. So please follow us also on Facebook and Twitter, and we hope to hear from many of you in our upcoming surveys. Thanks.
Thanks, Janet. We also have Sam Sadecki here from NECAN today. Sam, would you like to say a few quick words?
Yeah, hi. Can you see my screen all right?
Yep, Sam, it looks great.
Wonderful. Thanks for having us here today. I'm here representing NECAN. I am a co-chair alongside Jake Kritzker, who is the director of NERACUS as well. And so Emily Silva, whose contact information is our NECAN coordinator. And you can email her if you'd like to get on our mailing list and learn about our webinar series as well. We have a series of working groups that all work together, including an industry working group, an education outreach, a management policy, and a science working group. And recently we implemented an industry survey, and that has been an important tool for prioritization and engagement within our network. We're looking forward to using that as part of our monitoring to understand how to better co-locate these biological monitoring kind of moving forward. We'll be working over the next year or so on, kind of starting up a monitoring focused webinar series, and we were hoping to have a State of the Science meeting because we feel like we're a bit overdue, and hopefully we'll be able to work that out, obviously depending on all sorts of things that we're all working through in the world right now. But we're looking forward to hosting something along those lines in the next year as well. So just to give a little overview over some of the monitoring activities going on in the NECAN region, where we have co-located biological measurements alongside the chemistry. This is from a figure from a review paper that I led, talking about what to expect in the Gulf of Maine 2050. And in it, we had some figures of the current observational network for the Northeast, and the colors indicate kind of the frequency. So despite some of the spatial coverage, they're not occupied like as often as you'd like, but there is data there. And so that purple kind of goes from once every 4 years, and the red kind of indicates once every 15 minutes, right? So kind of spanning the scale of these, the cruises like the East Coast Ocean Acidification Cruise, which is one to mention on the NOAA monitoring front. It's going to be, there's another one going out this, scheduled to go out this August. So fingers crossed that happens. That, of course, spans everything from Canada all the way down through the Bahamas, and has a lot of co-located biological sampling funded by the Ocean Acidification Program. There's some EcoMon cruises that are funded by Northwest Fisheries Science Center, or by the fisheries, by North Fisheries. And those also have co-located, chemistry alongside the biological information. I'm highlighting a particular track here of the ECOA cruise that goes from UNH, the Coastal Marine Lab, out to along the Wilkinson Basin track, and that is part of the like an MBON Gulf of Maine project. It's highlighted in these light blue pieces, circled in the red. And then NECAN also hosts 2 buoys within Long Island Sound and the Great Bay, which measure ocean acidification parameters alongside nutrients and physical parameters as well. And so here's just highlighting a map from the EcoMon survey from 2021, showing you the chemistry stations along, because they're focused on observing the ecosystem more broadly. The chemistry stations are highlighted there in pink, so you can see where there would be co-located chemistry alongside the biological information from the EcoMon cruises. So that's a good resource in the area as well. NASA also does monitoring in the area, specifically the NATS transect, which goes in the Gulf of Maine, and that data is available, and that has been going on for like 18 years or so, maybe longer than that. So it's a long time series, with some of it including carbon chemistry. DFO also does some monitoring between across the Browns Bank and the Northeast Channel there. That has some co-located biological measurements alongside the chemistry. Also want to point out this monitoring acidification project, which has got the website there on the bottom. There's a GIS map that you can go into Long Island Sound and see all these different partners that are leading various water quality focused monitoring, which often has co-located biological observations with it. And then finally, citizen science activities going on. This one, of course, a big one was Shell Day, and the results and outcomes are discussed in 2 papers, one of them led by Jenny Rubin, and the other one led by Parker Gassett. And you can download these on our website under the references link, alongside the review paper that I mentioned of mine that's there as well, to find out more. But one interesting thing from one of the MOCA reports was highlighting this table talking about different parameters measured amongst community science programs within the 3 states of New England, in the Gulf of Maine. And in this case, what they found was that there was actually quite a bit of biological monitoring. So if you look at the biological monitoring route, you see that 78% of the citizen science activities had some biological monitoring in there, which far fewer of those have some carbon or other chemistry measures. And notably, there's some hurdles there, but obviously, kind of in an effort to co-locate, wanted to plant the seed. Thank you, Parker, for this, to think about, kind of augmenting our observations, especially in the nearshore environment. And with that, I'm looking forward to hearing what others have to say. And thanks for letting us give you an update from NECAN.
Thank you, Sam and Janet, for giving us the updates from NECAN and SOCAN. It's great to hear about some other work going on around the region in this area. And now today from NECAN, I'd like to give a warm welcome to Amy Maas and to Emily Rvest. Amy is joining us live from Bermuda today, an associate scientist at the Bermuda Institute for Ocean Science. Her research addresses questions about how the environment influences the distribution and biology of marine invertebrates. And her research on pteropod sensitivity to climate variables has taken her from the extreme environments of Antarctica to the Gulf of Maine and now into the mid-Atlantic. She's also currently working on a number of large interdisciplinary projects seeking to identify the role of plankton in global carbon cycles. Emily Rvest is an assistant professor at the Virginia Institute of Marine Science, William & Mary. Hi, Emily. And her research focuses on understanding how ecologically and economically important species like oysters, hard clams, and American lobster will respond to climate change. Emily's passionate about doing science that matters, often collaborating directly with members of the aquaculture industry. And she's also an avid science communicator, aiming to share the importance of her work with policy, industry, and public audiences. So today, Amy and Emily are here to discuss their research to develop bioindicators for ocean acidification in open ocean and nearshore waters, how co-locating chemical and biological monitoring can help identify OA tipping points and improve our understanding of ecosystem impacts in the mid-Atlantic, and how community programs like CSI Oyster can engage volunteers in biological monitoring efforts. So following their presentations, we'll have about 15 minutes for questions and comments. All participants will be muted today, so please ask your questions via the question box. And feel free to submit them at any time. Anthony Himes, our Sea Grant OA Fellow, will moderate the Q&A session for us as well. And just a quick reminder that the webinar is being recorded and will be available on the NECAN website, midacan.org, on the resources page. So without further ado, let's turn it over to Amy today.
Thank you. Let's see if I can figure out technology. All right, what do you guys see? Are we good?
You're great, Amy.
Okay, perfect. So I'm very excited to be here. I'm going to talk about some of my work with pteropods along the eastern seaboard. To give you guys just the background, I know everybody knows about OA, but let's just make sure we're all on the same page. Right, we add CO2 to the atmosphere by burning our fossil fuels. Some of that dissolves into the ocean, it changes the chemistry, and you get this disequilibrium. The part that's important to think of in the realm of the pteropods is that they have this really delicate shell. And so I'm going to be talking a lot about the saturation horizon. And everybody knows this, but when the saturation horizon is above 1, the calcium carbonate crystals stay together, and below 1, the calcium carbonate crystals fall apart. That's in the absence of any other biological or chemical activity that's going on. So those are just some of the terms that I'm going to use. And I'm talking about pteropods. So almost everybody who's thought about ocean acidification has heard about pteropods, but mostly we talk about these shelled species. These are the limacinid pteropods. And I think it's really important to know that pteropods come in a variety of shapes and sizes and sensitivities to acidification. And so the ones that we talk about a lot tend to be more polar or subpolar in origin, temperate species, but that they are found sort of throughout. And that I'll come back to that concept again a little bit later. The specific groups that I'm working on and talking about in this context are the species Limacina richiversa, which I'm showing right here. And the reason I focused on it is it does have an aragonite shell, so it's more susceptible to dissolution than things that have shells made out of calcite, right? So this is like baby shell for oysters, but these guys have aragonite their entire lives. They live in what we've classically referred to as vulnerable places, such as the open ocean, which has less variability in pH, or in polar regions where we know that the acidification is already causing things to reach tipping points. They are actually ecologically important as a prey item, as a grazer, and a contributor to carbon flux, because of those heavy shells, when they die or sink, they definitely sink fast. And the work that I'm going to focus on is work that I did when I was at Woods Hole Oceanographic Institution. So I was there until 2011 to 2015. And I was really focused on understanding coastal acidification in this region. So this is very much NECAN focused, where we were doing work in the Gulf of Maine region. Because the pteropods are so accessible there, relatively, what we would do is we'd drive up to P-Town, get on a boat, get out to Murray Basin, and then I could collect animals year round, and then use that to try to understand how OA stress lays on top of seasonality in the region. And in the Gulf of Maine, we know that pteropods are ecologically important for local fisheries. So this builds off of a bunch of different projects that we have going funded, but I'm also starting to try to translate that into a broader context, which is what I'll talk to at the end, which is a more recent grant from NOAA OAP. So the prior work, the like nitty-gritty science stuff, is we were going out on the Tioga. We would catch animals throughout 4 different seasons, and we would look at the carbonate chemistry. So here's our CTD. We had a little mini mock nest. We could find out exactly where in the water column the animals were found. And then what I would do is that I would bring animals back to the laboratory, expose them to various CO2 conditions, and see how they responded over time. So we have in situ, how are these animals doing over the seasonal cycle? And then if you add CO2, what happens to them? And the way that I was looking at this is I was using the respiration rate, so oxygen consumption, their shell quality, and I was also using gene expression, so transcriptomics RNA. I did not go too fast. So first, let's talk about shell quality. Shell qualities for pteropods has become a very common theme. We've seen lots of indications that their shell is strongly influenced by it. We see this both using really high technology, SEM, spend a lot of money, you can see the shell completely falling apart, or you can just put them under a dissecting microscope and shine light through them. And after only a few days, you see profound changes in what we're calling the opacity or transmission of light through the shell. So something is intrinsically changing in the shell microstructure that's causing them to become more opaque. Studies have also been done using dyes or stains that show how much a shell is growing or dissolving during exposures. As I said, I did 2 other things, oxygen consumption and gene expression. And the reason that I used those 2 things is that oxygen consumption is sort of like the paycheck you've got, right? So how much money you've got to spend. And gene expression is all the different things that you're spending that money on, right? So if you have enough food and you've got oxygen, you can burn that food and make energy. But that energy could go to different things at different times of years or under different conditions. The gene expression lets me look at where the energy is being spent. So in concert, these 3 things tell me a lot about how the organism is doing under the conditions of high CO2 and seasonally. So if I zoom back and I look at how all these animals are doing, remember there's a seasonal cycle. And in the Gulf of Maine, we have periods of pretty high CO2 already during the winter season. So more than the normal average CO2. And then in the spring bloom, when you've got all that phytoplankton in the surface water, you've got almost pre-industrial levels of CO2 in the surface water. These also are concurrent, however, with the food availability, because it's the phytoplankton in part that are making that change, right? They're photosynthesizing, taking in the CO2, putting out the oxygen. And so if I look at those just big scale metrics with the environmental variables, if you look at fluorescence, which is like our close version of how much food's in the water, how much phytoplankton, respiration correlated pretty well with food availability, with some extra bits that they probably don't just eat vegetarian. But respiration relates to how much food is available. Shell quality, on the other hand, seem to relate more to the aragonite saturation state we saw in the environment. Now, I talked about saturation state before and how the tipping point is at 1. Please note that we are seeing changes in saturation state above the level of 1. So we're seeing lower shell quality even at aragonite saturation states of 1.5. So when I take these animals and I put them in the lab and I'm like, okay, you've either been experiencing high CO2 or you've had no problem whatsoever. How do you feel if I torture you with more CO2? And this is the big zoomed out picture, is if we put the animals in different CO2 conditions, that's this aragonite saturation state. So this is least problematic, closest, this is ambient CO2 levels right here. And then I acidify the conditions based on that ambient level to what we'd expect in 2050 or 2100. If you look at the different months, 3 months out of the year, their metabolism is very, very constant and steady. Only in April during the spring bloom do they respond with an increase in respiration to high CO2. So here you put them in high CO2 and they have a higher metabolic response. Remember, this is the season when they had the highest metabolic rate to start with and they had the most food. If we look at their shells, it's a slightly different story. It's consistent, but still complicated. So what we have up here is that I'm looking at all the different months. So this is January, April, August, November, and then January again, because we did a bunch of sampling. And you can see that the slope of the line is the same always, which means if you put them in CO2, this is just for 3 days, that it does the same thing to them no matter what season it is. However, you notice that the starting point is different. And that's consistent with what I showed earlier. Their shells are reporting back what their experience was in the environment. So in January, when they'd been experiencing lower CO2, which is right here, aragonite saturation state of 1.5, their shells are reflecting that prior exposure. Then they come into the lab and experience changes in shell transparency over time. If I look at that, I have to get rid of January to then look at this so that it doesn't throw off everything. These are the days, how long I've exposed them to CO2, and this is the type of CO2 I've exposed them to. So here, they're hanging around 1.6, 1.5. This is the ambient condition for that time of year. It doesn't change that much over time. 3 days and 7 days isn't that different here. 1 day, 3 days, and 7 days isn't that different right here. But as the duration increases, you get an increasing opacity of shell. Now, this is important because when we talk about looking for indicators in the environment, we want it to tell us a simple story, right? How are you guys doing? But we're talking about something that's severity and duration interacting with each other. And when we see an animal, we only have a snapshot. And this is both good and bad, but when we try to interpret biological indicators, we have to keep in mind that it is a mixed message that's being sent in there. And when we design our experiments for biological indicators, we have to remember those two pieces, because the animals we observe are showing us just a snapshot, and we won't be able to tell which thing they experienced, duration or severity. The field data on distribution, the way that these plots are shown is you have how many bugs, and notice it's on a log scale. This is the depth, so we are doing this in Wilkinson Basin, which gets quite deep. And this is the blues are winter, the greens are the spring bloom, the oranges are like late summer, and then the purples are fall. And what we tend to see is that we have these blooms with the spring bloom, but that there's also sometimes a late summer bloom as well. And during those periods, the animals are found very high up in the water column. In the winter, they tend to have a bimodal and deeper experience of the environment. We know that this is a spawning event, both of these, because there's substantially smaller individuals that are responsible for these peaks. So we can track the phenology of the reproductive events. They're spawning twice, once in the spring bloom primarily, and again in the later summer. And that's important because when we think about when these animals are spawning, we know for sure that those juveniles are super, super, super sensitive. They're more sensitive than the adults. We know this because we did experiments to see exactly how sensitive the babies were. So what these charts are is this is the ambient condition, 400 parts per million at the time, and this is our high condition, 1200 parts per million. The lighter color is the less developed, the darker color is the more developed. And as we track the development of these individuals through time, we see that under normal CO2, we get greater development and we get our only velager. And in our high CO2, things are delayed. This is further complicated by the fact that there's more mortality in the higher CO2. So we think that that transition from stage to stage, which we know to be highly energetic, is also causing greater mortality. So we know that these animals are producing babies at 2 times a year, and they do it in the very, very surface water. This is important because when we look at the biological sampling, those vertical components are not always co-located for biology and chemistry. The big picture that we get from this highly technical science part is that the metabolic response does seem to be tied to the spring bloom and associated reproductive trends. And it seems to be trying to favor the timing of those reproductive trends, which makes sense. You make babies when there's food and when they're not going to die immediately. Their shell condition was consistently and distinctly influenced, but it varied among the seasons, which reflected that natural exposure, which is great because then we can possibly use that as a tool. Their gene expression, which I haven't shown here, showed patterns of seasonal responsiveness to CO2, naturally, reproductive cycles, and lipid storage. And there's some potential CO2 biological indicators, both in the environmental data, but also in the exposure data. So we see that the early life stages are the most sensitive, but they're not naturally found during periods where acidification was present in the surface water. And I'm showing that here. This is from Wang et al. 2017. So this is May. This is the only time of year we were seeing any of the waters in the Gulf of Maine going under saturation, but it was in the benthic zone, okay? So all those babies are happening, but they're happily up at 50 meters. And so they never experienced that undersaturation in the water column. And you say, okay, that's cool, but who cares about pteropods? And it's not like we eat them. But they are good bioindicators because they show signs of acidification stress before other organisms, and they don't die. Like, they can hold a shell despite the fact that it's a little frosted and keep on swimming. And so then what we can do is use that to look at what other things are going on in the environment. What we really realize is that the when and where of exposure matters, especially because of the early life stage piece. If you happen to be a juvenile, I don't know, decapod crustacean or juvenile scallop, it may actually matter that after the spring bloom, the deep waters are acidified, because those larvae don't stay in the upper water column. They eventually need to go to the benthos. So figuring out the phenology of when those transitions occur and where the co-located places and periods of stress are going to be is really critical. And so our argument has been for a while that we need better coupling between biological and chemical observations at those specific life changes and in the correct portion of the water column, so that we can identify danger zones for populations of greater interest. One of the ways that I've worked collaboratively to try to turn this information, which is, again, very specific to the Gulf of Maine and a bunch of critters that very few people even know, I mean, who knows how to spell it, is that we worked as a group to do something that's called threshold analysis and consensus building. So all the people who study pteropods got together and tried to make all of the experiments, which were done with different species in different regions with different experimental exposures, and tried to figure out, okay, when do we truly think these organisms are first identifiable as responding to ocean acidification? And so what we have is the saturation state and the specific metric that we're studying. And one of the ones that we kind of coalesced around was this one, so that we see mild dissolution frequently after 5 days. And so bunches of studies, 5 days-ish was about the time that people were seeing it. Sometimes people saw it before, sometimes people saw it after, but because the experiments were not designed the same way, this was allowing us to come on to something. And so using that 1.5, and again, remember, that's an aragonite saturation state way above what we've flagged as problematic. At 1.5, we see that there's a response to acidification in Limacina. So what can we do with that? Okay, so what we want to do with that is be able to say, if pteropods are affected at 1.5, can we identify places and times that might be environmentally problematic for other organisms? And what we can do, this is output from a regional model. So it takes the hydrography and the air and the currents and the chemistry and all the data available. And this is in the California Current Monitoring System. And it takes that threshold of 1.5. And it says, okay, everywhere where it's white, you're above the threshold of 1.5. If you're these colors, this is the deviations below that, okay? So you fall below that 1.5 threshold. As you get warmer, it gets worse. Then we also have the duration piece. This is how long you spend below that threshold. And this gives us that interactive term that I was talking to you about. It matters both the severity and the duration. And this gives you hotspots, okay? These are places where both the duration and the severity interact, that you would think that the ecology of that situation might start to be negatively influenced by acidification. And it lets us pinpoint places, and then you can look at how that co-varies. So this gets to the project that I'm currently working on. We've been trying to take advantage of those EcoMon surveys that Samantha talked about, and they have a bunch of biological monitoring, but they were designed for fisheries studies forever ago. And so they're doing counts of all of these organisms, and what we're trying to do is add just this tiny biological monitoring into the system so that we can then say something about it. We also are adding surface to deep carbonate chemistry every time we find pteropods. So not just at the carbonate chemistry stations. So getting our sampling into the workflow of the standard monitoring is what we're trying to do. And then we're trying to make it like, keep it simple, stupid monitoring, right? Like anybody who's never seen a pteropod before can grab that pteropod, put it on a microscope, take an image, analyze that image, and get a repeatable and consistent version of the data so that we can add that into monitoring much more easily. That turns transparency into an additional environmental factor when we're trying to do our analyses. And then we can overlay that data onto what we are measuring and predicting for the region using our models and observing data. So what we're trying to do, and this project was funded during COVID, right before COVID, so it's been tricky, but what we're doing is I want to take the abundance of things that we actually do care intensely about and use them with my factor to create community level analysis of regions and seasons in the Gulf of Maine based on this pteropod indicator when they're caught concurrently, and then take the environmental variability to predict places amenable to those species of interest or not amenable based on our analysis. And so the way that we put that together is we put all the data into NCEI, and this is more important than you think. Getting it all into one place is really, really valuable, because then we can start to see the co-location amongst all of these different sampling regimes. And then we synthesize this data with gridded DIC data, and this is leveraging both NECAN and SOCAN's data sets. And then the goal is to translate this information into the state of the ecosystem reports. My big, grand someday goal is to add my index and tipping point analysis to the data portals, so you could overlay how the biological and chemical data work. And so this is showing you a map that was from the NECAN data portal, and the image is from Grace Saba. The blue is the current OA sampling, and again, it has that resolution component that Samantha was talking about. And the colors are the scallop fishing effort. And what I think would be really valuable is if you can generate specific drop-down menus where you could see the data and suggest regions that would be at risk based on both severity and duration of acidification, based on our understanding of what the chemistry of the water color is doing. And for more frequently sampled sites, this can also be presented in a more like weather channel version, right? You can have literally plug in an equation. If we're getting continuous measurements of the water column, how much the water fell above or below our severity and above or below our duration, and then create that ongoing throughout the year, see which times of year animals are consistently exposed and for what duration. Obviously, we need to link portals like this to regional models with forecasts and hindcasts, because I think that's what people really need for management. So I always feel like I need to come away and tell people that the world is not ending exactly yet, and what we can do about it if we are concerned about the trajectory. And I think good investments that we're doing right now is that we are continuing to monitor populations and increase the observation of acidification near our marine sources, which is great. Everybody's working together to get our prior data together. There's been a lot of joint efforts to synthesize data sets so that we can start building predictive models. It is critical to keep modeling future habitat ranges of both the commercially important and bioindicator species. And we do still need to run sensitivity experiments on regionally important organisms to plug those into their models, because pteropods behave very differently than fish or shellfish. And even though we have specific information of different organisms, it is valuable to increase that, keeping in mind the sensitivity and duration piece. It is important, however, I think, to discuss reasonable local action, even on non-OA fronts, to ease thresholds and model effectiveness, because we do need to place policy that addresses the big picture questions of resiliency, sustainability, and renewables. So actually truly get at the question of CO2, as well as allow local ecosystems to implement resilience building policy in the face of things that can't be helped on a local basis. And so with that, I want to thank you for your attention. This was obviously something that's been going on for a number of years with billions of helpers. And here are the papers. I think that you all, if you can't have access to them, please do email me. I'm happy to send them. And with that, I think we move on.
Hey, someone please shout if the audio or visual isn't working.
You're perfect.
Thank you. So thanks so much for having me today. It's also a pleasure to follow Amy on this series on biological indicators and linking biological and chemical observations. And the theme of my talk is going to be slightly different, to focus on how I currently think about this in the context of community science. And in the presentations today, you've heard folks refer to citizen science and community science, and I just wanted to clarify that community science here, the term that I'm using has historically been called citizen science, but I choose intentionally to call it community science because US citizenship is not a requirement to participate in the work that we do. So I hope you'll consider broadening that, your use of that as well. So for acknowledgements, there are so many people that have contributed to this work as a community science project. There are many, many participants. In the current iteration of the funding for the work that I'm going to share with you today, this is in collaboration with Drs. Jill Bible and Sarah Clark DeReza at Washington College in Maryland, and Sarah Beam and Emily Beck, who are high school teachers in our respective states. In its current iteration, the work is funded by NOAA's Ocean Acidification Program, and this community science program is currently in its 5th year of data collection, funded iteratively through a variety of ways, initially by the Dominion Foundation Environmental Education and Stewardship Grant. And then there have been many contributors to this work, including a variety of graduate students at VIMS, including Anthony, who's on the call today, and staff at VIMS. And then this project has also provided a lot of opportunities for undergraduates at William and Mary and other schools for gaining some hands-on field experience. So Amy introduced acidification, and now that we're moving into the coastal environment, wanted to also talk and introduce coastal acidification. And both of these acidification processes result in changes of the carbonate chemistry of seawater that have similar symptoms that affect coastal marine organisms. So as Amy mentioned, ocean acidification is primarily driven by anthropogenic carbon dioxide that is absorbed by the ocean, whereas coastal acidification has other drivers. One example here that I'm using is nutrient runoff from the land that stimulates algal blooms. And when those blooms subside and that organic material is degraded, carbon dioxide is produced that adds to acidification, compounded with whatever CO2 has been absorbed from the atmosphere. And both of these processes that increase the amount of dissolved CO2 in the water modify the carbonate chemistry system that generate a couple of chemical symptoms that coastal marine organisms are sensitive to. We see decreases in pH and decreases in saturation state. And Amy was talking a lot about aragonite saturation state. And for the adult organisms that I'll be focusing on, I'll be talking about calcite saturation state, but they're very similar. So another thing that's important to keep in mind when thinking about acidification in whatever realm of the ocean you work in is the frame of reference that you're using. And this figure here is from the 2021 IPCC report that shows our current record of global ocean surface pH, as well as various projections into the future from different climate scenarios. And our average ocean pH is currently about 8.1. When we think about Chesapeake Bay and where I do a lot of my work, our baseline is very different. We are on average around 7.8. So we already have a different starting condition in terms of 2021, 2022 levels. And then we think about how additional injection of CO2 from atmosphere or our coastal acidification drivers will affect that. In this SSP5-8.5, this most severe atmospheric CO2 scenario, open ocean pH is projected to decrease to 7.65. And if we apply that same offset to, or a similar offset to our Chesapeake Bay conditions, we are off the scale of this plot all the way down to an average of 7.2. So when thinking about coupling these conditions, these kinds of measurements, it's important to have this framework in mind of we're looking for ocean, the signature of ocean acidification as impacts. What are we really looking for? Now, as I mentioned, ocean acidification and coastal acidification are known to affect marine organisms. When thinking about oysters for this talk, some work out of our group has illustrated some of these impacts. Others have found this too. So this is an example with eastern oyster larvae from a PhD student in our group, Annie Schatz. And we can start to visually see impacts as early as 2 days old in these larvae. You can see in that lower picture, the shells look more uneven and crinkled, and they are on average smaller. With adult oysters, we've done some work to look at rate processes. So this is an example from a study where we measured rates of net calcification of large and small adult oysters. And to orient you to this plot, on the x-axis is omega calcite, with the right side being a scenario of no future acidification, and the left side being scenarios that are much more acidic. And so you can see for values that have a net calcification that's positive, those are oysters that are growing through time in a positive direction. And then scenarios where there's a negative net calcification, these are scenarios or individuals where the shell is dissolving overall, dissolving more than it's growing, which is pretty frightening. And so the lines here represent if we were to average across all the individuals that we measured, this is the relationship between omega calcite and net calcification rate. And you can see that there is, and I don't see my mouse working here to highlight this, but there is a point just above about 0.9 omega calcite where for the small oysters, that green line crosses the 0 y-axis threshold. So we could identify that as a tipping point or a threshold where those small oysters are going from net growth to net dissolution. Now, this was done in the laboratory where we were only manipulating carbonate chemistry, but these controlled experiments can be very helpful in identifying metrics, or in this case, calcification as a metric that we could measure in the field to look for evidence of impacts of acidification in the field. So why, in this case, is it really useful to couple biological and chemical measurements in the field? This is because acidification is not happening uniformly across the landscape. It's occurring across a mosaic of natural conditions. And so from a single open ocean measurement or something similar, you wouldn't be able to anticipate from one oyster farm to another, or restoration reef to another, or one tributary to another, what acidification is going to look like. And so here are some examples of this natural mosaic. From Margie Friedrichs at VIMS, on the left-hand side, we have a map of modeled conditions for bottom water pH from March 3rd, 2021. And from all the different colors you can see, there are a variety of different projected pH conditions spatially throughout the bay during that time, which varies by 0.4 to 0.5 units. And a lot of the space is oyster habitat. Similarly, in the time series plot on the right, this is a 30-day representative time series of near surface water pH near VIMS. And the colored lines on there show for open ocean, present day in purple, and end of century in red, conditions of pH. So you can see that on a day-to-day basis, pH is swinging dramatically. And this is another reason why it's important to have this informed framework for thinking about ocean acidification in the places where you want to know impacts. So given this very complex landscape, how can we best monitor ocean acidification and its impacts? So for linking biological and chemical observations, there are several critical needs that I think about in this scenario, and these aren't, there are many more than these. They're just a few to inspire you today. So what water conditions do organisms and communities actually experience? A lot of the chemical observations that we've been made have historically intentionally been done away from strong biological influences on that carbonate chemistry. And so if we want to know how an oyster is going to experience ocean acidification, we need to know what water conditions that it actually experiences. So we need to be taking measurements on oyster reefs or in the habitat of other species of interest. How variable is the natural environment? What kind of data do we need to detect ocean acidification and its impacts? So understanding that landscape will help you figure out how can we actually detect the signature of ocean acidification and tease apart the direct impacts of ocean acidification. Along that vein, what biological metrics can allow us to separate the impacts of ocean acidification from impacts of other environmental variables? So Amy mentioned food as something that was also had a relationship with the respiration rate of the pteropods. And so can we find metrics that allow us to look at sort of the net effect of the environment on the animal, but also ones that are sensitive only to ocean acidification if we're really interested in documenting the independent impacts of acidification. And of these metrics that we could use, which can be easily measured by a variety of data collectors, if we're thinking about leveraging community science in this effort, and I'll use an example from Amy's talk, someone being able to take a picture of a pteropod shell that doesn't require a very fancy instrument or something like that. We can gather so much more data that way. And then the bonus here is if these metrics are predictive or representative of community or ecosystem level impacts. So when I think about a biological indicator and looking for a canary in the coal mine, you know, we would love to have the early warning system where we could take one measurement and be able to stop the train from going off the rails before it does. But that measurement would then need to be predictive of that train going off the rails. So here's the opportunity of community science. One huge advantage of community science is that you have many people who contribute to this work, and that allows you to do so much more, to collect data in more locations, to sample more distant sites than you could manage on your own, similarly to survey a wider range of environmental conditions, and to engage more people in ocean acidification research, which is always a wonderful thing. So with that, I'll transition to talking about the community science program that I started here. With this goal, when I started it, to really expand our footprint of understanding of carbonate chemistry variability in the Chesapeake Bay region and how that relates to oyster ecology. It's called Community Science Initiative Oyster, or CSI Oyster, and we work primarily with high school students and their teachers with the purpose of monitoring impacts of variation in water quality, inclusive of carbonate chemistry, on oyster growth and survival over time. And so in this iteration of the project, we are partnering with a high school in Virginia, Chesapeake Bay Governor's School, and a high school in Maryland, the Gunston School. And on the map here on the left, the green dots are our current sites and that are active in this round. The other sites identified with the black dots are ones where we have that we have used historically but aren't active in the current project. And these sites we established with the teachers, and they and their students visit the sites every 2 to 4 weeks to measure the survival and growth of oysters that we've provided them, and to measure various aspects of the water conditions. And the goals for this particular project are to deepen high school students' understanding of the scientific process and its role in addressing complex ecological problems, advance environmental literacy and public awareness about ocean acidification, and improve a community science data collection program that integrates water conditions and oyster ecology data. So beyond, so we sort of added value to this general data collection program by also having these educational objectives. So with this project, we engage about 100 high school students every year, as well as multiple undergraduate students and graduate students in my program here at VIMS. There are 3 faculty members currently on the project, and then we also have summer students and other volunteers that participate. In the center photo here, you can see how we deploy the oysters at each site. And every time that the students visit the site, they will measure the oyster parameters and the water parameters. So we provide them with a handheld YSI instrument that they use to measure temperature, salinity, and dissolved oxygen and pH. And we've also trained them how to collect and preserve bottle samples of water using best practices, and those are returned to us at VIMS where we measure pH and total alkalinity. For oyster size measurements, they measure the longest dimension of the oysters or shell height using calipers, and they also take photos of each individual oyster for later analysis using ImageJ. One of the most profound impacts of the project on the students comes from taking these photos, and this is just a sample time series of photos from oysters at our Urbana Creek site where you can visually see how much they're growing over time. The students from taking these photos can anecdotally tell the difference between oysters from the different sites and notice that they're growing over time. And so it's a great way to make the data more tangible for them. It's very impactful. So to get into some of the data, I don't intend for you to read through all this table, but it is summarizing our water conditions at the different sites. And throughout the 1 year of data collection, our most recent complete year of data collection, which was 2020 to 2021. And I wanted to highlight some patterns for you. So we have our 7 different sites as rows, and then the different water conditions as columns. So if you look at that calcite saturation column, you can see that on average, we have some sites where average saturation state throughout the year is below 1, and others where it's around 1 or above 1. And one of the benefits of this kind of data collection is that you can look at trends in the carbonate chemistry with respect to other variables too. So for example, we have 2 sites that have low saturation state and lower temperature, and another that has a similar low saturation state but a higher temperature. So we can look at the role of temperature at low saturation state. Similarly, with our higher saturation state sites, we see a large range in salinity. And so we have the opportunity through this natural experimental setup to look at the relative influence of salinity and carbonate chemistry. So these were just a few examples of how this kind of data set could be used to get some of those critical needs that I mentioned earlier. In terms of the biological data, the plot on the right here is showing shell height at the end of the 1 year deployment. So the oysters all started out at a similar size at all the sites. And at the end, we can see that in general, the oysters in Virginia, the most right-hand 4 sites are bigger than the ones in Maryland. And so we can consider what environmental factors, including carbonate chemistry, might explain this pattern in growth between the sites. And there are multiple contributing factors: water chemistry and temperature, salinity, and dissolved oxygen. But to summarize the trends, the general trends with you, that the sites with higher growth tend to have higher salinity, higher omega calcite, and therefore better calcification conditions, although there is a lot of nuance in this. And with the 5 years of data, once that's complete, we're planning on doing a more sophisticated analysis to try to tease this apart. So stay tuned for that. This project has also provided additional opportunities to add value. So this was an add-on led by an honors student at William & Mary who worked with me And she took the oysters from the CSI Oyster project at the end of one of the years and put them on common garden conditions for a month, which is means they were all experiencing this. They were previously at the 4 sites. Then for a month, they were all experiencing the same conditions. And she looked at how various traits changed over that 1 month as a way to tease apart the role of proximal and distal environmental history on their physiology. So this left-hand panel is showing you that at the end of the CSI Oyster deployment, condition index, or the ratio of tissue mass to shell mass, was lower from oysters at VIMS Pier than the other sites. But you see that over the common garden period, condition index decreased in the oysters from those 3 sites, whereas it increased at VIMS Pier. And she wanted to see what water parameters at the distal sites, so observed during that CSI Oyster period, explained these patterns in condition index. So she looked at omega calcite salinity and temperature and found that salinity was the only significant predictor of those 3 of the change in condition index. So in this case, although omega calcite did differ between the sites, it was salinity that was really the most important driver of this particular physiological parameter. Another way that we're using these data is through modeling efforts with Margie Friedrich's group. So Faye Daw, one of the PhD students in that group, has used our CSI Oyster data, incorporated that in an effort to develop salinity-alkalinity relationships for various different tributaries in Chesapeake Bay. And that has allowed us to estimate total alkalinity for the YSI-based time series that otherwise don't have full carbonate chemistry associated with them. So that has allowed us to do some really cool modeling, analogous to some of the things that Amy shared, where we're looking at time series collected in various different tributaries of Chesapeake Bay, and from the carbonate chemistry and integrating our biological threshold information, calculating the hours per day that an oyster at that location would spend below its threshold. So in this case, for the York River, the figure on the right, you can see as we're going up the tributary and salinity is decreasing, so to the left on that plot, we see that the oysters are spending more and more time below that threshold, with spring popping out as a season where there's relatively more exposure below that ocean acidification threshold at a particular location. So this is my last slide. I wanted to summarize by sharing the critical contributions emerging from this work, this kind of work. One, that we're getting a much better idea of the diversity of habitat conditions, both spatial and temporal, in the present day. It's allowing us over time to document and predict the emergence of impacts of ocean acidification and other kinds of environmental change in our area. And there is a role for this kind of community science in being able to help us tease apart when and where ocean acidification is a defining ecological driver, and when is it more of a threat amplifier, but not the primary variable that these organisms are responding to. And it will also be very helpful in allowing us to identify bioindicators that are reliable in situ in the natural environment outside of a controlled laboratory experiment. And then, of course, there are many benefits of engaging community members in this work. You can enhance the ocean acidification literacy of the public. They allow us to capture more spatial and temporal data, and they allow us to understand how these bioindicators operate or translate to different settings, for example, a restoration and aquaculture settings. So with that, I'll thank you very much, and hope we have some time for questions.
All right,
Thanks
great.
so much.
Oh, sorry, go ahead, Kirsten.
No, go ahead, Anthony. You're great. You can take it from here.
Okay. Thank you both Emily and Amy for your talks today. We do have some questions that have already come in, but anybody else in the audience that would like to continue submitting your questions, please feel free to do so now. So our first question that came in was for you, Amy, and it's, can you clarify a bit about the duration of the exposure? How long are we talking about, days, weeks, or longer?
Yep. So from the lab experiments, we start to see changes in the shell as quickly as 3 days, right? But I was keeping them alive for up to 2 weeks in the lab, and they were still flapping around. So the natural exposure then could relate to any period within that context that they feel it naturally. So because the lifespan of these guys is, I assume that it's limited by lifespan, and I also have a vague impression that it's probably changing over the duration of the life of the organism. Otherwise, we wouldn't be seeing that variability among seasons. That's within one lifespan, unless they're different critters, blah, 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 blah. Short story, I can prove between 3 to 12 days, I don't know longer. But it is enough to tell us sort of a hindcast of the environment. I hope that helped.
Yeah, I think so. Our next question, also for you, Amy, asks, are the effects you are seeing reversible under an emissions reduction scenario? And if so, on what time scale?
So for the individual organism, we do see evidence that the pteropods can fix their shells. So then remediation, if they were to come into contact with water that had been ameliorated by some sort of human activity, whether that's growing seagrass, whether that's some like engineering solution, I think that they would have the capability to respond to that. In the broader time scale, these animals have experienced seasonal cycles in their CO2 throughout their history. That's part of their natural environment, their normal phenology. And so if we started tipping everything back down the other way, that would absolutely help. But both things being true, yes, I think that local human impacts could have important implications for these guys, but because they are open ocean organisms, we just gotta stop burning that fossil fuel.
Okay, great. Our next question is for you, Emily. Do you think it would be possible to add some phytoplankton sampling to the high school sampling? This would be a great addition to see how the food sources compare.
Yes, that's a great question, and that's one thing that we added with this recent iteration. The challenge was to think about how to do that in the context of community science, so something that the students could measure. So the students don't have, since they're in the field, they don't have access to a microscope, or they're doing this after school hours. And so what we added are Secchi tube and Secchi disc measurements as a very loose proxy of chlorophyll A, but I think we could follow that up with more paired measurements. We also tried to use, there are certain apps that allow you to look at watercolor, so we thought that would be a cool way, paired with the Secchi measurements, to try to get at food availability. And if anyone has any ideas for other things we could try, I would love to hear them. We found that based on different light conditions, it was too hard to compare the color of the water with the glare off the water and things like that. So again, in that field setting, it didn't end up working very well for us. But yeah, that's something we definitely want to try to capture, and just trying to figure out the best way to do it in a community science context.
Great. We have another question here for you, Emily. Can you again describe and elaborate on why you think salinity was the statistically relevant parameter in the grad students' analysis that you showed? I think they're referring to Jill's paper.
Yes, thank you. Well, certainly oyster physiology is sensitive to salinity. It's known to to be a driver of oyster growth and physiology. It just hasn't been looked at as closely with respect to carbonate chemistry. So I thought it was interesting that it did emerge as the dominant driver, or the only significant driver in that case. But I think, given that we only had 7 sites, it would be really interesting to look at that over more years of data, or over a wider range of conditions, to see when and where that is the primary driver. So, and certainly there could be certain salinity ranges or certain ranges of saturation state where we would see ocean acidification emerge as the primary driver. So that's one of the things that we'll be getting at in a similar but different way in this longer term analysis of our time series. Yeah, it's a great question.
Okay, this next one's for you, Amy. Can opacity of pteropod shells be determined if the samples have been preserved with formalin?
No, unfortunately, no. There's actually some really good papers that look at the different preservation techniques, and even with buffered formalin, it does mess things up over time. Even ethanol, which we originally thought no problem, apparently you also have to buffer ethanol. I learned that a very hard way. So unfortunately, going back into historical records gets really quite difficult with the pteropod shells, but I think that's also a warning for us to consider when we can think about going back into the historical record for calcite organisms as well. So sad, but onward and upward. Now we're doing a better job of buffering.
Great. Emily, this one's for you. Is the pH determined with handheld YSIs accurate enough to calculate calcite saturation?
Yeah, we don't use the pH from the YSI handheld to calculate saturation state. We measure pH from the preserved bottle samples using the spectrophotometric method, the best practice. So we use that to calculate saturation state. But the pH is a redundant measurement, but also gives the students something to look at immediately in the field as they're visiting the different sites, so they can see the relative difference in pH, even if it's not measured to the accuracy we would use for the carbonate chemistry calculations.
Okay. This is a question, I guess, that kind of applies for both of you, as you showed saturation state thresholds that are different than one. But do you guys have opinions, I guess, on how much that may vary species to species? Do you think it would be consistent, or is there a lot of variability there that you would expect?
Oh, it's going to vary. Right. So it not only is it going to vary, like life stage is going to vary depending upon which mechanism by which an organism is affected. Right. So like if you've got a non-calcifier, if you've got a fish who's got otoliths, but like has more control over their pH balance, it's going to vary. And so again, it's really tricky to like find the magic bullet one number. And that's why this idea of having biological indicators that tell you, okay, well, for our most sensitive species, we're crossing a threshold. We don't necessarily know that that's going to immediately result in a threshold being crossed for all organisms. But since we're trying to understand ecosystem function, or at least that's what I'm hoping for us to consider, is that if one member of the ecosystem is suggesting that it's starting to be in peril because it's a web, because it's a connected network of organisms, that to be conservative, we look at the thresholds of the sensitive organisms. And then to be less conservative, when we're like, okay, now everything's really on fire, we can pick different thresholds. And so I think it's like an awareness that above one can still matter for very sensitive things is really important there. But again, there are going to be times when we need to have more like specific thresholds for, should we start a remediation effect in this particular basin? Yes, that should be done with a different threshold. Does that make sense? Emily, your opinion.
Yeah, it is definitely going to matter, or it's definitely going to differ between species and life stages and populations even. Part of it is because the saturation state threshold of 1.0 has been used as this convenient threshold, but it really refers to this thermodynamic threshold rather than a biological threshold. And so, as Amy said, depending on what parameter of carbonate chemistry that physiological process is responsive to, it might not be concentration of carbonate ion in the water. So you might see that biological tipping point occur at a saturation state that's higher or lower than 1. One example from the work that we've been doing, looking at various different life stages of the eastern oyster, is that we've found that these thresholds often occur below 1.0. Even in terms of calcification. And with calcification, there's so much biological intervention in the process of calcification that those organisms can still be, creating an artificial environment in that calcification space, even though the exterior environment has a saturation state less than 1. It takes a lot of energy to do that, but it is possible in certain cases. And so with eastern oyster, we found that the tipping points that had been identified for the Pacific oyster and the West Coast hatcheries and the crisis doesn't apply to eastern oysters. They're much more resilient from what we've looked at so far. And so we're in the process of identifying species specific and even aquaculture line specific thresholds for oysters in our area.
All right, great. Thank you both very much. I think that's all the time we have for questions today. So I'll turn it back over to you, Kirsten, to wrap us up.
Thanks so much, Anthony. Thank you, Emily. Thank you, Amy, so much for sharing your research with us today and really talking about the challenges and also the common threads in trying to monitor these two very different systems. I thought that was really interesting to kind of hear what you're experiencing in both those different worlds and where those commonalities might be too for management. So I just wanted to let everybody know that the webinar recording will be available on our website at necan.org shortly. So thanks so much for joining us today and have a great afternoon.
Hi, everyone. Thanks for having us.